Greetings. Um, so let's go ahead and get started with the immune system. So, what's the role of the immune system? So, the role of the immune system is to clean debris and repair. Debris is stuff that's not good, that the body doesn't consider good. So, what are some of the examples of debris? So, it could be things like toxins, foreign molecules, or pathogens. And pathogens can include things like bacteria, fungi, parasites, and the list can go on and on and on. Viruses, it could be something that could be internal, for example, tumor. So, again, if it's a cell that's become cancerous, that's also debris, that's also bad stuff. And part of what the immune system wants to do is get rid of it. So let's review some important terms. So let's kind of talk about some important terms here. Self. So what's self? Self is part of our body. Um, so it could be our cells, it could be organs, it could be organ system. What's tolerance? What's tolerance? Again, immune system recognizes self or recognizes part of your body and it tolerates it. It doesn't attack it. It's not supposed to. So that's tolerance, and that's a good thing. You want your immune system to recognize you and tolerate it and not attack it, and you want your immune system to recognize debris or things that are not good and not tolerate it and attack it and get rid of it so that the body can heal itself. So that brings us to the interesting point of non-self. That's not part of our body. That's foreign. That's what the body considers harmful. I want my immune system to identify things that are non-self and get rid of them. Let's talk about antigen. Antigen. It's another term that's thrown out. And the hope is that once we get a sense of what the terms are, that the rest of it becomes easier. Makes sense, right? So what's an antigen? So antigen, the simplest way to think about it is a marker. So it's a marker. So how can I think through this, or what's an example that can help me learn? So I consider antigen a tag, luggage tag. Okay. And the body that it's tagging is actually the suitcase or the luggage. Okay. So, it could be a bacteria, which is luggage, and then you have a marker on that bacteria, and that's an antigen. You could have a cell, that's your body cell, and there's actually an antigen on top of that, and it's a marker. Okay. So the suitcase is your body cell, and the antigen is the marker or the tag. Okay. Now, from an immune system perspective, there's something to be considered. For for a marker or a molecule to be considered a marker, it's got two aspects it needs to have. One is it needs to be immunogenic. So it needs to be involved in one way, shape, or form with immunity or immune system or immune response. And the second is it needs to be reactive. So it needs to elicit some kind of immune response or not elicit some kind of immune response. So it needs to have a role to play within immune response. And it needs to elicit an immune response, whether it be, yes, I'm going to kill this, or no, I recognize this is my own body. Okay. Let's talk about antibody, right? So I think that this is another concept that tends to confuse people. Okay, so how do I think through this? So remember luggage? When we talked about antibody, we talked about a suitcase. So this is our luggage. This is our suitcase. Okay, and then this is the tag. So this is antigen, right? So this is suitcase, and it could be a body cell, my cell, or it could be bacteria, or it could be anything else. Okay. What's tag? It's antigen on this suitcase. So it's got my address, my name, and that's how you recognize, hey, this suitcase belongs to Prashant. Okay. Now, what is antibody? Antibody. Antibody. Essentially, it's a protein against a certain antigen. Antibody. Anti against body. Okay? So I have my suitcase and I have a tag, okay, which is my antigen. What antibody does is it's a protein which fits in perfectly with this antigen tag. Okay? So that's what an antibody is. That's the simplest way to think about this. Okay? So again, we're going to discuss more about the role of antibody, but the biggest role of antibody, or the overarching goal of antibody, is it is able to hook into antigens on cells, and it's able to identify these antigens and mark them or flag them. And by virtue of doing that, when it flags them, it tells the body, hey, that there's something going on here. You know, whether it be good or bad, but there's something going on here. Okay. This is the way to think about it. So let's say it's my suitcase, right? So the suitcase has a tag on it, which is the antigen. It says Prashant, and it's got my address, it's got my phone number. So what the antibody does is it attaches to that tag and flags it for the immune system or the airline people and say, hey, we need to do something about this. Okay? We need to do something about this. So we're gonna talk a little bit more later about antibodies and how they work. Okay. Next, we're gonna talk about our cytokines. Cytokines. These are essentially chemicals. These are essentially chemicals. What do they do? They are involved in coordinating immune responses. Okay. And essentially they're produced by cells, and the cells can be infected with bugs or they can be not infected with bugs. Okay, so cytokines, chemicals, they're involved in coordinating immune responses, and they're produced by cells. Okay, it's as simple as that. Chemokines, on the other hand, are also chemicals. They are also involved in immune responses. And they are a type of cytokines. They are a type of cytokines. So chemokines are a subset of cytokines, and they are produced by cells that are infected. Okay, so that's the difference. Is cytokines are produced by cells, and these cytokines are produced by cells. Cells can be healthy cells or infected cells. Chemokines are cytokines. They're a type or subset of cytokines that are produced by infected cells. Okay? So the next thing to look at are the soldiers of immunity. These are actually the immune cells. Okay? These are the immune cells. So remember we talked about chemicals that help us coordinate, cytokines and chemokines. And then we have soldiers that actually do the work. And they are white blood cells or WBCs. Or the prefix that we use is leuco. Okay? So if you have sites, right, as suffix cells, leukocytes would be white cells, white blood cells. Okay? Easy, right? See, names make perfect sense when you think through this. Okay, cytosis, increased number. So, increased number of white blood cells, leukocytosis. Pinia, little, decreased number. So, what's decreased number of white blood cells? Leukopenia, leukopenia, decreased numbers of white cells. Poiesis, to create. Poiesis, to create. So the process of creating white blood cells, leukopoiesis. Leukopoiesis. See? Terms that make perfect sense. Make it easy for us to learn. Okay? And we're going to use this paradigm a whole lot. We're going to talk about types of cells. For example, neutrophils. So if you have decreased number of neutrophils, neutrophils plus penia is neutropenia. Okay? Easy, right? So that's a few terms right there. So let's talk a little bit next about dividing immune system. Okay? Let's figure out how to divide this immune system. Because if you divide it, it makes the study much easier for us. So there's two ways to divide the immune system. So again, there's two divisions. Okay, there's innate immunity and there's adaptive immunity. So innate immunity, this type of immune system, this section of immune system, this part of immune system, does not differentiate between pathogens, between debris. It doesn't have different pathways or specific things for specific organisms. It's what I call a hammer. Okay? It boom, it hits. And it takes care of business regardless of what the enemy or the debris is. So it doesn't differentiate. Adaptive, on the other hand, is smarter. It differentiates between bacteria 1 and bacteria 2, virus 1 and virus 2. It creates different tools to take care of bacteria 1 and different tools to take care of bacteria 2. Okay, so that's difference 1, big difference. Secondly, innate immunity does not need priming. It's immediate. Why? It doesn't need to think. It's immediate. Doesn't need priming. Adaptive immunity needs a bit more time because it needs to study what the enemy is, create tools to fight the enemy, and then fight it. Okay? So again, difference between innate and adaptive immunity. And it's amazing how innate works with certain pathways, adaptive works with certain pathways, and how innate works with adaptive and adaptive works with innate to actually take care of the debris or take care of the enemy and help the body heal and repair. Okay. So the next thing to consider is, hey, who are these troublemakers? What's the debris? Okay. So one can divide this into two parts. The external troublemakers. So you have bacteria, parasites, fungi, toxins, foreign molecules. These are all things that stay outside the cell, and they're external troublemakers. Now, the second type are troublemakers that create trouble inside the cell. So viruses actually get inside the cell, and now the cell is the trouble is in trouble and is creating trouble. If a cell becomes cancerous or it undergoes dysplasia and starts to change or morph, now that cell is trouble. Okay? So that's the way to divide the troublemakers. And why do we need to do that? Well, so here's the deal. So remember immune system? We have the innate immunity and the adaptive immunity. Remember? Innate immunity does not differentiate, is immediate, does not need priming. Adaptive needs priming. It does differentiate and it does need time. So innate immunity does not differentiate. So whether it be external troublemakers or internal troublemakers, it does not care. I don't care. It attacks. Adaptive immunity, on the other hand, differentiates. So, hey, I need to know, are these internal troublemakers or troublemakers that got inside the cell now and they're creating trouble? Or they're external troublemakers? Okay? Because remember, adaptive immunity differentiates. If my own cells are troublemakers, or something has gotten inside my cells, and now those cells are being used to create trouble, I have a different pathway for them within the adaptive immune system. And that's the cell-mediated immunity. Why? Because my cells are in trouble. Okay? My cells are in trouble. So I'll need something to mediate and help me get rid of these cells or take care of these cells. Okay? Cell-mediated immunity. Okay? And again, we're going to study this a bit more in detail, but again, that's the T cytotoxic cell pathway. So like I said, we're going to study this a little bit more. And then again, you may have an external troublemaker. Okay? And when there's an external troublemaker like bacteria, fungi, pollen, right? Now that's a different pathway. That's humoral immunity. Okay? And that uses B cell and the B cell pathways. And we'll talk a little bit more about it. Okay? So again, let's just kind of go over it one more time. So remember immune system, you had the innate immunity and then you had the adaptive immunity. Innate immunity does not differentiate. Adaptive immunity does differentiate, and it is subdivided into cell-mediated immunity or humoral immunity. Cell-mediated immunity takes care of things where the cells are either in trouble, so like cancer or dysplasia of those cells or how the cells change, or if the viruses get inside the cell and now they're using cells to create trouble. So again, these are my cells that are being used to or are creating trouble. So I use cell-mediated immunity. So that's first part of the adaptive immunity. If there's external troublemakers, then it's humoral immunity. So those are the two subparts of adaptive immunity. Okay, perfect. So now we have a good idea about immune system, how it's divided, different kinds of troublemakers, a little bit of terminology, went through these, and what the role of the immune system is. Okay. So let's start studying the soldiers of immune system and their origin story. So like all the superheroes have their origin stories. Same way, um, so does WBCs. Why do I need to know this? Well, why do I care? It's very interesting. When we start to look at diseases, we're going to look at big terms like multiple myeloma, acute myelocytic leukemia. And the reason this is important to cover this portion is because if you understand this, uh, that aspect is going to be very, very easy. You're gonna breeze through it. It's gonna make perfect sense. If you don't know this, then it's a compilation or hodgepodge of things which don't make any sense. So you can't think through it. You can't stack knowledge on top of each other and increase your knowledge base and apply that knowledge and use critical and clinical thinking. So let's look at leukopoiesis, or how these WBCs are created, and what are these WBC cell lines. Okay, so again, all of it starts in bone marrow. So we're going to start here. So you see this? Okay, so all of it starts in the bone marrow. Okay, and then again, there's some cells that go to thymus gland, and then it goes to the blood vessels. Okay, and then it goes to tissues. So that's how I've broken it up. Is again, you have three steps. All the WBCs get generated in the bone marrow. Some of them mature in the thymus. They go into the blood vessel, and then they get into the tissue where they act. Okay. Okay. Now, within the bone marrow, we start with a pluripotent stem cell. So this stem cell is the original superhero, and it can differentiate into different types of cells and give us what we need. So the way it does is it differentiates into two cell lines. One of them is a myeloid stem cell, and the other one is lymphoid stem cell. Very, very important. Myeloid and lymphoid. Okay. Then within the myeloid stem cell, I have my liver and kidneys. They produce TPO. It's an hormone, an enzyme. Okay. And that helps the myeloid, stem cell differentiate into megakaryocyte. And mega is what? Huge. Wow, these are huge. Okay? Huge. Huge. Mega karyocyte. Easy way to remember it, right? Now, it's so big, it's gotta get out in the blood vessel. And again, we have blood vessels, which is the sinusoidal blood vessels that go into the bone. Okay, and so they've got these narrow spaces where the cells can squeeze out of. Well, this is such a big cell, squeezes out of these little spaces. Okay, and breaks up, bursts into platelets. Aha, I get my platelets. Okay, easy, right? So myeloid stem cell, TPO enzyme produced by kidney and liver gives us mega, huge karyocyte, huge. And that bursts while trying to get into the blood vessels and gives us platelets. Again, myeloid stem cell, okay, that is acted upon by erythropoietin or EPO, different enzyme. Remember TPO? Now this is EPO, erythropoietin. Again, EPO is produced by the kidneys, and it differentiates into erythrocytes and gives us red blood cells. And we'll talk a bit more about this process when we look at hematology, which is or the hematology system. Okay, but this is where the money is. Okay? This is where the money is for the immune system. I give this, I introduce a granulocyte colony stimulating factor enzyme in there. Okay? A granulocyte. And it gives me a monocyte. Now, because it's an agranulocyte colony stimulating factor, stimulates the production of monocyte. Yes? So monocyte is a agranulocyte. Okay? Monocyte is an agranulocyte. There's no granules. And it's going in the blood vessel. Okay? So that's why you can get a count. You can get a monocyte count. Because you can take somebody's blood and get a monocyte count. Now, it goes through some processes and it can then migrate into tissues where it becomes a macrophage or dendritic cells. Okay? Macrophage or dendritic cells. So what's the difference? Well, one of them is in the blood, the other one is outside the blood. That's the difference. All of them don't have any granules because who created them? A granulocyte colony stimulating factor. So that's monocyte for you. Okay? Easy. Now, the macrophage, remember, it's now in the tissue. So macrophages can be in different organs. If they're in bones, they're called osteoclasts. In lungs, it's alveolar macrophages. In brain, it's microglial cells. And so on and on and on and on and on. But the concept is very simple. It is an agranulocyte, and it comes from the monocyte line. Okay, and that's coming from the myeloid stem cell. So it's coming from the myeloid line. Okay, easy. And phage, phagocytosis, phage, eat, likes to eat, likes to eat. So likes to eat, there's no granules. Okay, easy. Next, now I introduce granulocyte colony stimulating factor. Okay. So remember I gave it agranulocyte stimulating factor. Now I give it granulocyte stimulating factor. Okay. And when I do that, I get 3 cell lines. And because they are being stimulated by granulocytes, stimulating colony stimulating factors, they are all going to have granules in there. Easy, right? That's correct. So again, the granulocyte colony stimulating factor gives us a precursor cell, then gives us a band cell, and then it gives us the actual cell. So immature, getting mature, mature. Okay? And it's the same for all 3. So you have basophils, eosinophils, and neutrophils. And I call them the Ben cells. Ben, B-E-N, Ben cells. Makes it easy for me to remember. Okay? So again, you have the bands. Now they're still immature, but they're getting mature. And then you have basophils, eosinophil, neutrophils. These are all have granules in them. And we'll talk a little bit more about these granules. Okay? So again, basophils, eosinophil, neutrophils. Now they are in the bloodstream. So what else is fancy about them? Okay? Well, they can actually get into the tissue, basophils, eosinophils, and neutrophils. Okay? Okay. Additionally, there's something very interesting about basophils, eosinophils, and neutrophils, other than the fact that they have granules. Their nuclei. You know how the nuclei is like this, right? Their nuclei is multilobulated. Okay? Multilobulated. See, difference? This is round. This is multiple lobes. Okay? So they are also called polymorphonuclear cells. Poly, multiple, morpho, morphology, how they're shaped, right? These different lobulated nuclei, okay? Polymorphonuclear cells, cells with multiple morphology of their nuclei. So they are abbreviated as PMNs. So basophil Ben cells are PMNs, polymorphonuclear cells, and they have granules. Well, within the tissue, the basophils become mast cells. So they mature even more, and when they mature, the lobulated cells become more round and they lose their polymorphology of their nucleus. So they don't have they're not PMNs anymore, but they still have granules. Okay? Just some things to think about. So again, you have monocytes. Remember, we are still on the myeloid stem cells or myeloid lines. So you have monocytes because they're stimulated by agranulocyte colony stimulating factors. They don't have granules, and they become macrophages and dendritic cells, and they don't have granules either. When you give granulocyte colony stimulating factors, you have basophils, eosinophil, neutrophils. All of them have granules. And also, they've got this nuclei that is lobulated and has different shapes. So they're PMNs. And basophil becomes mast cells. So they do have granules, but by then they've matured and they don't have their nuclei is got a more definite shape. Okay. Now we're going to continue. Okay. Let's talk about the next line, which is, so remember we were myeloid cells, which is we've got the monocytes, the macrophages, and the dendritic cells. Okay. And then we've got the Ben cells, and then the Ben cells, the basophils also become mast cells. Ben cells are also PMNs. They've got granules. Mast cells are not PMNs. They've got granules. And then you have lymphoid stem cells, a different line, right? Different from myeloid. And when I give them a granulocyte colony stimulating factor, and that's the key, a granulocyte, I actually get some more immune soldiers. Now, are they going to have granules or are they not going to have granules? So they're not going to have granules, or at least the granules that we can see. Okay? So these wouldn't have granules, and we actually get three cell lines. We get natural killer T cells, we get B lymphocytes, and we get T lymphocytes. Okay? Those are the three cell lines we get. Okay? You have natural killer cells, which go into blood and go into tissue. You have the B lymphocytes. They mature in the bone marrow. And by maturation, what the immune system wants to do is wants to make sure that they have the right receptors and that they are not immunogenic towards self. So if you start to release B cells that uh, starts to target your own body, it's going to eat itself up. It's going to tag the body incorrectly. So part of maturation is to make sure that doesn't happen. And then they get released into B lymphocytes, and then they go into lymph circulation. Okay? And there's different types. There's naive B cells, there's active B cells, there's memory B cells. Okay? And they produce antibodies with the help of T helper cells. And we'll talk a little bit about them. But this actually forms the humoral immunity. So remember we talked about that adaptive immunity, okay? We talked about adaptive immunity. When it's an external troublemaker, we use humoral immunity, and we actually use this part of the immune system, which is the B lymphocytes, which give us B memory cells, B active cells, B naive cells, and antibody. And all of this is humoral immunity for external troublemakers. Okay. Remember, this is still the lymphoid line. Okay. Okay. So let's look at the last portion of this, which is the T lymphocytes. So again, we are giving the lymphoid stem cells a granulocyte colony stimulating factor. So again, T lymphocytes, they're created in the bone marrow. However, they are do not mature in the bone marrow. They do not mature in the bone marrow. B lymphocytes mature in the bone marrow. T lymphocytes do not. They mature in the thymus. And again, they are part of the adaptive immunity. So we want to make sure they are not adapting to attack the body. So in thymus, they go through the same process, some of the same similar processes to make sure they have the correct receptors and they are not immunogenic towards self. Okay. Essentially, from thymus, they go. These T lymphocytes go into circulation, especially the lymph circulation, where there's a type called T cytotoxic cell. That's the big one for cell-mediated immunity. Okay, so remember we had the adaptive immunity. Within that, we had these troublemakers that are my own cells that are making trouble. So this actually constitutes cell-mediated immunity. Okay, and so these are the immune cells. So the question is, well, why do I need to know this? Why do I need to know this? Let's just kind of zoom out just a little bit. Okay, let's just zoom out just a little bit. And let's just see why we need to know all of this stuff, right? And just go over it one more time. So again, we talked about the myeloid and lymphoid cell line, right? That's coming from the bone marrow. Why do I need to know this? Multiple myeloma. Well, right off the bat, myeloma. It's going to be related to the myeloid cell lines. Multiple. It's going to be multiple cell lines that's affected. Oma, cancer of this particular tissue type. See, makes it easy, right? If I say acute myelocytic leukemia versus acute lymphocytic leukemia. Now you know myelocytic leukemia is myeloid cell line and lymphocytic leukemia acute will tell you the course of the disease. Lymphocytic will tell you it's the lymphoid cell line, and leukemia, malignancy of the white blood cell. So it can help you figure out what the disease is and what's going on. Okay. So we talked a little bit about myeloid. Myeloid cell line gives us platelets through megakaryocytes, gives us red blood cells through erythrocytes, and gives us monocytes and macrophages and dendritic cells, all of which do not have egg granules. Okay. And then again, the myeloid cell line gives us basophil, eosinophil, neutrophils. All of them have granules. And basophils become mast cells in the tissue, and they do have granules. But basophils, eosinophils, neutrophils have this lobulated, many-shaped nuclei, which mast cells lose, and they just get a very definite shape for their nuclei in the tissue. So Again, basophils, eosinophils, neutrophils, mast cells, all are granulocytes, but only basophils, eosinophils, and neutrophils are PMNs, or polymorphonuclear cells. Okay? Why is that important? Well, you'll see terms like granulocytosis, PMNs. Okay? So now you know what are granulocytes. Okay? And last but not the least, we go into the lymphoid stem cell line where you get natural killer cells, B lymphocytes, and T lymphocytes, and they're all, they don't have granules because they've been stimulated by the agranulocyte colony stimulating factor. And so when you talk about agranulo cells that don't have granules, agranulocytosis, that means too many cells that don't have granules. We're talking about TB, natural killer cells, monocytes, macrophages, and dendritic cells. Versus granulocytosis. Now I have basophils, eosinophils, neutrophils, and mast cells that are increased in number. See, makes perfect sense, makes it easy, right? And so you have natural killer cells, you have B lymphocytes that mature in the bone marrow, get in the lymph circulation, and B lymphocytes are part of the adaptive immunity that fights against external enemies, humoral immunity, and then you have T lymphocytes that fight against um, internal enemies or enemies within the cell, so like cancer cell and tumors and viruses which get inside the cell. Okay, so again, if you see here, adaptive immunity, so you have the B cell, which is the humoral immunity, okay, for external troublemakers, and then you have T cell lymphocytes, which are for cell-mediated immunity, they're for the internal troublemakers, and these two form adaptive immune cells. This is adaptive immune system. That's it. That's the adaptive immune system. Rest everything is innate immune system or innate immune cells. And that's really, really important to know. Okay? So again, innate immune cells are everything other than B and T lymphocytes. Okay? So what about basophils? Yeah, they are part of innate immunity. Why? Because again, they're right here. They're not B and T lymphocytes. Okay. Easy? Easy. So now we've seen the cells, and now we've seen how the cells go from one space to another. And the way we've divided the immunity was innate immunity and adaptive immunity. In adaptive immunity, we divided into humoral and cell-mediated immunity. Well, guess what? We've actually divided all our soldiers and categorized them. Now, there's a couple of things that are pretty interesting. You guys see this natural killer cell? Okay. Which cell line is this? It's the lymphoid cell line, right? However, it is part of the innate immunity. It is part of the innate immunity, and it's innate immune cell. Okay. However, which cell line is it belonging to? The lymphoid. Remember, lymphoid had three cell lines: natural killer cells, T natural killer cells, B lymphocytes, and T lymphocytes. Well, other than natural killer cells, all the lymphoid stem cells, all the lymphoid cells, or cells of the lymphoid line, are involved in adaptive immunity. So natural killer cells are the only exception of the lymphoid cells that are lymphoid but are not adaptive. Okay? Okay. So just very interesting stuff to note. All the myeloid cells are innate immunity. So the easy way to remember is B cell is humoral immunity, attacking external things. T cells, and they are part of the adaptive immunity. T lymphocytes, cytotoxic cells are cell-mediated immunity, and they're again part of adaptive. Rest everything else is um, Rest everything else is innate immunity, which includes all the myeloid cell lines, all the myeloid WBC lines, and the T natural killer cells. Okay. Okay. So again, if you're talking about polymorphonuclear cells, what are these? These are the Ben cells, basophils, eosinophils, neutrophil cells. What about granulocytes? It's all the Ben cells. They've been stimulated by the granulocyte colony stimulating factor, and then mast cells. Mast cells comes out of basophil, so they have the granules. Their nuclei matures and just stops having many, many shapes. What about agranulocytes? All the monocytes, right? And then the monocytes have macrophages and dendritic cells, and then B and T cell lymphocytes and the T natural killer cells. These are all agranulocytes. Okay? So these are the cells. These are our soldiers and how they're organized. Okay? And that's immunity is essentially a study of soldiers and chemicals and how they interact with debris. Okay? So let's talk about the next thing here real quick, which is, okay, so now that I have an idea about the soldiers, how how do I know? How do I know what is self and what's not self? Okay, just give me a couple of seconds and then let me get to the right screen here. Okay, and we'll get going. So that's the next question to kind of tease out, which is how does the body recognize self, right? How does the body recognize self? Right? Because if it is not self and it is considered a threat, now I can have innate immunity and adaptive immunity go after it. If it's self, is it good self or bad self? And if it's good self, I gotta leave it alone. If it's bad self, I gotta destroy it, right? But we have to recognize whether it's us or not, because if it's not us, chances are it's a threat. Okay? So essentially, so there's two parts to this. So all nucleated cells in my body and in our bodies have a receptor on the cell surface, which is called MHC, major histocompatibility complex 1 receptor on the cell surface with a self antigen. So what does that mean? That means, so this is my cell, okay? This is my wonderful, beautiful cell. This is my nucleus with my DNA in there. Essentially, there's an antigen which this cell takes in, which is a self-antigen, or it's part of my body. And then the nuclei creates a protein which is called MHC1, and it gets to the surface. And at the surface, it is now having two things. It's got a receptor, which is MHC1 receptor, and it's got on that receptor little self-antigen. And we have it in all our nucleated cells. Okay. Now there are three genes A, B, and C, in one of our chromosomes that help us with this particular antigen, helps create this antigen, that helps us create this receptor on our cell. Okay? And this receptor with the self-antigen basically tells the cells in our immune system, hey, it's me. I'm your brother. I'm your friend. Let's not fight. I'm one of you. Okay? Now, what if a virus gets inside the cell? It's very, very interesting. Let's take a peek. Okay? So here's a little virus. Okay? And it got inside the cell. So what does it do when it gets inside the cell? It does a lot of things, but on the simplest level, what it does is it tries to incorporate its uh, genetic material into the nucleus and into the cell machinery and create proteins. And a lot of those proteins are going to be replicated viruses. So it creates proteins which then actually tag on to this self-antigen. So this is MHC1. And now you have a self-antigen. So this was the self-antigen. And this is the additional part which the virus introduced. Now, when the immune system looks at it and the immune cells look at it, and we'll talk about T cytotoxic cells because that's what they do, they say, hey, this ain't right. This ain't good. He's not my friend because something changed. Something changed, and this is not a self-antigen anymore. There's something else going on here. So there's a change in the major histocompatibility, one receptor, and that is tagged, that is read by the immune system and recognized by the immune system as a threat. So that's how viruses, even though they're inside the cell, they're still they're still recognized, and the cells that they're part of are destroyed so that they can prevent using body's own cells to damage the body. The other thing is, hey, I what if I become cancerous? So again, I'm a cell. I have my nucleus. I have my MHC1, and I have my self-antigen, right? So what happens? This cell is now becoming dysplastic and starting to go towards cancer cell. Well, it may be that it may lose its MHC receptor on its cell surface, and the MHC1 could be downregulated, or the MHC1 has changed. And guess what? If there's a change that's detected again by our adaptive immunity and our cytotoxic cells, and they tag it and they destroy it. So again, makes a lot of sense. I love MHC1 because it helps me figure out who me is. Okay? Now, there are three exceptions to this rule. Or they're not exceptions, but there's an additional rule that's added on to them. And these three cells are um, cells that are immune cells, and they have not only MHC1, but they have another marker, which is MHC2 marker. And those three cells are dendritic cells, macrophages, and B cells. Remember B lymphocytes? That's the lymphoid line. Yes. Okay, so that's the B cells, lymphoid lines. And then remember the monocytes and macrophages and dendritic cells. So again, that's from the myeloid line. They don't have granules. And so it's the macrophages and the dendritic cells. So these are three cells. They have another marker in addition to MHC1, and it's MHC2. So here's those three cells, B cell, macrophage, dendritic cells. They have their MHC1 and they have their self-antigen, same as this one, because they're nucleated. And then they have this MHC2 marker. Okay. So what if macrophages get infected by viruses? Well, they're going to undergo the same changes that we had here in this area. They're going to undergo the same changes, and they're going to get tagged and destroyed. What if they become cancerous? Again, they'll undergo the same changes they did in this area. Makes sense, right? Okay. Okay. So that helps us make sure they're in line. They don't become troublemakers. And if they do, we gobble them up, we eat them up, we clean them up, we treat them as debris and clean it up. But this MHC2 receptor is very interesting. So what happens is this part of the immune system allows these cells to present antigens to the adaptive immunity. Okay? It's a very interesting concept. So what happens? Antigens, remember what are antigens? These are these markers. So these protein markers. Okay? So you have antigen, okay, that gets inside the cell. Okay? Again, recognized by the nucleus. The nucleus creates this protein which attaches to the antigen and then gets it to the surface. So now on the surface you have MHC2. Attached with that, there's an actual antigen, and it's presenting the antigen to another part of the immune system. We'll talk a little bit about it. Okay, so these three cells not only have MHC1, they have MHC2. Now there's additional three genes that control this MHC2. They're DP, DQ, DR. So remember ABC for MHC1, PQR for MHC2, but it's got a D in front of it because remember MHC2 is 2. So the genes are labeled with 2 letters. And that's the way that I remember it. Why do I need to know the genes? Not really. The big thing is to know that MHC1 and MHC2 have 3 genes each that control these markers on the cell. So when we are doing transplants, we got to try to make sure at least these 6 genes are matched between donor and recipient. And we'll talk a little bit more about this when we talk about transplant, because that increases how the recipient, how the graft holds in the recipient. But we'll talk a little bit more about that later. But essentially, you have these 3 genes, and this is MHC2, which sits with the antigen and presents this antigen to another part of the immune cell. And there are 3 cells, B cells, macrophages, and dendritic cells that do this. They show to the immune system the external threat when activated. Okay. All right. So that's how we differentiate our body cells from other body cells, and clinically what their role is. Okay. So again, to recap, what did we talk about? We talked about the role of the immune system. We talked about some terms, some basic terms. We talked about soldiers and chemicals of the immune system. We divided the immune system into innate and adaptive, and we subdivided adaptive into two parts. The cell-mediated immunity, which is for my own cells, like for tumors and viruses, which uses a T cytotoxic cell pathway. And then adaptive immunity, which is humoral immunity, which is for external troublemakers. Okay? That's very briefly what we talked about in part one of this. Part two of this, we talked about leukopoiesis. We talked about myeloid and lymphoid cells. We talked about monocytes and how they become macrophages and dendritic cells. We talked about the Benz cells that have granules and are PMNs, and then mast cells that come from basophils that have granules but they don't have PMNs. These are all part of innate immunity. And then you have the lymphoid stem cells, which become T natural killer cells. So again, T natural killer cells are lymphoid, but they are part of the innate immunity. And then you have B and T lymphocytes, with B lymphocytes maturing in the bone marrow and T lymphocytes maturing in the thymus. And the B lymphocytes are what are responsible for the humoral immunity for the external troublemakers, and the T lymphocytes, especially the T cytotoxic cells that are responsible for the cell-mediated immunity, and they both form they're both part of the adaptive immunity or adaptive immune cells. And then finally, we talked a little bit about how the body recognizes itself, which is the MHC1 receptors, and how it is attached to self-antigen, and how when viruses and tumors come in, how they change it. And then we have MHC2 uh, for 3 cells, which is the antigen-presenting cells, B cells, macrophages, and dendritic cells, that again, when antigens come in, they create an MHC2 receptor on them with the antigen on top, and they present the antigen to a part of the immune system, and then that gets processed, and we'll talk a little bit more about it in the subsequent videos.